My name is Josip Blonder, and I'm going to give a seminar on mass-based clinical proteomics in biomarker research. I am a head of clinical proteomics at the Laboratory of Proteomics Analytical Technologies at Frederick National Lab for Cancer Research. So first, I will give you some introduction to clinical proteomics, then talk about mass spectrometry-based biomarker research, what are the challenges, what are the limitations, what solutions we can offer, and where we applied it, and finish with concluding remarks. So, clinical proteomics can be defined as the application of mass spectrometry for molecular profiling of clinical specimens in all aspects of clinical research. As you can see from this slide, we use tissue specimens regardless whether it is laser capture microdissection, whether they are thin tissue slices, fresh frozen tissue, formalin-fixed, paraffin-embedded biopsy samples. We also look into the body fluids, serum, plasma, urine, cerebrospinal fluid. We also use different kinds and different modalities of liquid chromatography that include strong cation exchange, affinity depletion, size exclusion, and use also high resolution and high accuracy mass spectrometry to develop custom bioinformatics and using advanced data meta-analysis. So why bother with clinical proteomics? There are several reasons for that. First, that allows for direct measurement of matured proteins, which are final products of gene expression cascade. So the proteins are direct bioeffectors and they are found closely at the center of pathological or biological process. Then also, recent proteogenomic studies show that significantly higher correlation between levels of RNA and corresponding proteins than previously thought, so that gave more significance to the proteomic efforts, at least for the gene and proteins regulated closely at the transcriptional level. However, there are the genes that there is significant mismatch between RNA and gene product proteins at the proteome level, and there are several reasons why that is, and we know that is existing. There are post-transcriptional and post-translational regulatory mechanism, and also that there are other things which why clinical proteomics is important, that we have samples like body fluids, special types where genomic analysis is not very helpful, and also for the cell like blood cells and platelets, they don't have RNA, so the proteomics that comes as a very clinical proteomics is a very useful tool. And the other reason why you would like to look into the clinical sample is that data obtained from cancer cell lines of animal models are not always predictive of what is actually happening in human cancer in vivo. So we were able to very efficiently cure cancer in petri dish in animals, but really we are not as successful as in humans. So these discrepancies between model and human cancers and in vivo and vitro system, the affected ability of researchers to devise more effective cancer therapeutics. So it is important to have a look directly into the clinical specimens. So what are the other, why bother with clinical proteomics is also that unlike transcriptomics, expression proteomics can measure differences in protein expression in subcellular compartments. So the study that we did long time in 2005 using global profiling of lipid drafts and O18 or 16 labeling to look into the iota B toxin internalization in a cell line. So that was really agnostic proteomic study, and that study revealed that basically CD44 is 3 times upregulated in iota B treated cells, and that was one of the candidates that we suspected that might be facilitating iota B internalization into the cells. And last year, our collaborators were able to do series of biological experiments, and they have shown that really CD44 facilitates the internalization of iota B. So that is an example how the proteomics can be very useful, that genomics most probably couldn't do anything about it. And obviously that paper was published in PLOS and showing the CD44 promote intoxication by the Clostridial iota B toxins. The another thing is also that another study to exemplify importance of proteomics we have done with a group in Canada. We have looked into the HUX cells that are treated with the LPS lipopolysaccharides, which is important for the sepsis outcome. And we have found in that study that SASH1 is a protein which is important in regulating LPS response and including TLR receptor. So that is important also that unlike transcriptomics, expression proteomics can facilitate mapping of post-translational modification, and we have also shown that this protein is ubiquitinated and it's also making complexes with TLR4 receptor. So those are two examples to show that really without proteomics one cannot move further. And then the another thing is also, which is important, which we would like to, I would like to pay attention, what kind of the samples we are going to use. Clinical proteomics workflow is complex. Specimens acquisition, storage, sample preparation, multidimensional preparation. It's much more greater number of dependent experimental variables than if you're doing cell lines or lysogenic cells or transgenic animals. The question is there, what are the samples that one would like to use? There is a huge amount of formalin-fixed samples. They are good, they have good availability, and it is a big push to use those, but we still believe that fresh frozen samples are the golden standard, because you can one can do proteomics, genomics, and metabolomics at the same level, which you cannot do with formalin fixed. And then it is also good immunoreactivity and unbiased proteomic quantitation, which is difficult to do with the formalin fixed tissue. So anyhow, we would recommend using fresh frozen, although there are a lot of successful studies that are using formalin fixed tissues. So we have applied our approach to show how successfully we can look into the very small amount of samples, and we have investigated mouse model, transgenic mouse model of cardiac hypertrophy, which was triggered by tetracycline-based manipulation of HRAS. We have basically done a very simple thing. We have used 8-micron thin slice using methanol-based digestion and solubilization, and compared the proteomes of hypertrophied heart and normal heart. And obviously the heat map shows that we have been able to capture the differences between those two tissues in the protein expression level, but this is not the ultimate goal. The ultimate goal was to see what is biology that is driving that transformation. So nonetheless, it's important to say that we were able to identify HRAS in a tissue. Obviously, it was confirmed with immunohistochemistry and Western blotting. Subsequently, we did the pathway analysis, and we have seen, we were really excited to see that we are covering EarthMap signaling, which obviously is downstream the RAS, and in your left corner you can see that p-value is extremely high, but it means that it's important that the analysis is capturing the biology, because if you use any kind of, and one use any kind of pathway analysis, you will get something, but p-value is extremely important to say how enrichment of that pathway is important in your sample. And obviously that was also confirmed with Western blotting. We do believe that every proteomic analysis has to be cross-validated, and we have also seen that the phosphorylated ERK is also upregulated in HRAS when HRAS is constitutively signaling. The other important thing is also that HRAS signaling is basically forcing myocytes to produce embryonic myosins, and we have shown those two myosins in Westerns, which shouldn't be shown in a heart after the birth, because heart exit the dividing cycle after the birth of any mammal. So these embryonic myosins show that when heart is stimulated with HRAS, you have some kind of mesenchymal activation there. And the most important thing is also that pathway analysis show us that beta-catenin VNT pathways are activated secondary to the HRAS activation, and we have also confirmed that using immunohistochemistry. So that story tells us, this is an example how you can use a tissue proteomics to not only detect the differences but find the pathways which are driving that process. So our conclusion was that VNT pathway has been activated secondary to the HRAS activation. One other interesting thing is also that since we have found that after constitutive HRAS activation, we have VNT activation and basically the differentiation, because the myocytes which are changed based on HRAS signaling really look like embryonic cardiomyocytes. Completely independent on that, there is also studies of the scientists who are looking to develop stem cells and turn them into the cardiomyocyte fate to treat the myocardial infarction, so that tissue is not replaced by scar tissue but viable myocytes. And they also found that Wnt inhibition is important to differentiate pluripotent stem cells from cardiomyocytes. So that is something that we can show that proteomic analysis can be used even further to hypothesize some and trace some other biological effects of inhibition of Wnt pathway. So now I would like to turn into the biomarkers, why we do need them. I mean, definitely the majority of cancer treatments are still categorically aside, and you can see the Kaplan-Meier survival curve, and this is the only way how to judge the treatment. This is the only biomarker basically that help for ages clinicians to tailor the therapy, and those are just different protocols, and if any gain on survival time is there, then the treatment is changed. But this is definitely trial and error, because aggregate population statistics is applied to an individual diagnosed with cancer. If you don't have better biomarker, this is a good way to go, but we obviously, using that kind of the treatment, you have basically, if you have the patient group diagnosed with the same kind of cancer, generally, if you do the error in trial and this Kaplan-Meier curve-based treatment, basically there are four possible outcomes. Drug can be toxic and not beneficial. Drug can be toxic but beneficial. Drug, it doesn't have any effect, and only one small patient groups have that the drug is not toxic and beneficial, which is in your lower corner. So basically, 3/4 of patients most probably do not have favorable outcome. So because of that, we need to have the biomarkers. So that is, clinical proteomics is one way to try to discover, basically, to match diagnosis and treatment strategy to each individual with unique molecular wake-up. So ideally, it should be based on systemic use and information about patient molecular genotype, phenotype, and metabolic profile to tailor optimal therapeutic care for given individual. It's basically matching the drug with the patient. And one cancer, and in biomarker, ideally, if you have a biomarker which drives the pathology and it's a drug target, this is ideal story. So breast cancer is definitely 10 years in front of all the other cancers, because we have good biomarkers and we can do the subtype of breast cancers like a HER2 positive, progesterone and estrogen positive, or triple negative. So at least there there are 3 different subtypes, and the treatment is completely different. Like in your right corner, you can see in A is HER2 positive breast cancer and HER2 negative, and Herceptin is a monoclonal antibodies treatment for that. So here we need to be aware that is really happening. It's a paradigm shift. It is shift from organ-based to pathway-based oncology, and really development of drug targeting selected molecular pathways actually. And that luxury of breast cancer, the other cancer doesn't have. So that effort is proteomics one-way, clinical proteomics, that we can address that issue. So I would like to show this slide as a paradigm that we have basic biomedical research and research in clinical setting, and what people are typically doing in this way. We have animal model cell lines, and we move our discoveries from branch to bedside. I really do believe, we really do believe that there is another part of that circle, that the story is full and the circle is closed, that we also can use from bench to bedside to bench, because we have human model and clinical samples, and then all picture is basically completed. So there are 2 parts of translational aspect. One is from bedside to bench, other is from bench to bedside, because whatever discoveries can be made using clinical proteomics, they can be checked on well-defined animal models or cell line. But initial discovery should be done at a place where pathological process is really happening in vivo, when we have all the micro environment plus cancerous cells, because this we do know that those things cannot be separated. So how we are going to go with the biomarker discovery? Typically, the question is whether one would use tissue or body fluids. If you use body fluids like a blood, the benefits, it's easy to obtain, but there are some drawbacks, because the biomarker in blood are diluted, it's low concentration, they're outside of its cell environment, and we don't know where it's coming from and what happened to it along the way. While in tumor tissue, this is difficult to obtain, but one can, based on discoveries on the tissues, which most probably has higher local concentration of biomarker at the site of the process, and you can study within the cellular microenvironment, and we can also compare to the normal tissue and do mechanistic studies. So in editorial that we wrote a couple of years ago, we basically decided one shouldn't do one or other. The best thing is to use it both, so that you can use tissue-directed approach. And we wanted to see and hypothesize, can we really detect tumor proteins in peripheral blood? So using shotgun proteomics. And that was the study we published a couple of years ago. We have used renal cell carcinoma as a model disease, and you can see that this right kidney, although both of those kidneys have the same genome, the right kidney has a tumor growing in, and that is the why do we need what is happening in situ at the level of that tumor. So what our experimental design and why we use renal cell carcinoma is also because it is easier to remove a kidney rather than other organs. Renal cell carcinoma arises from the cell of proximal tubules. It's histologically is a clear cell type, and its incidence is rising, and an estimated 50,000 individuals were diagnosed in 2007, while 12,000 died of that cancer. So it is still an important cancer to look at, although it doesn't belong to the group of big four: colon, prostate, breast, and lung. So our idea is basically to compare normal tumor tissue versus tumor tissue, and take peripheral blood before the surgery. So because during surgery some biomarkers can leak into blood, and basically this is what we call it is tissue directed approach. Then we have to analyze at the same time 3 samples from the same patient on the individual basis. So basically this is our experimental design. And we call it like tissue-directed identification of disease-specific blood biomarker using subtractive expression proteomics. So we would analyze tissue and peripheral blood in the way that we would look at normal adjacent tissue and tumorous tissue, and then do subtraction and look only into the proteins that are identified only tumor. Not because these overlapping are not interested. We believe that proteins that are identified by mass spec only tumor might be really extremely biologically important. And then overlap it with the preoperative plasma and see how many of those which we identify only tumor we can see in peripheral blood as potential biomarker. So that is another way of showing it. We would do the tumorous tissue, normal adjacent tissue, preoperative plasma. Since we are going to use spectral counting there, it is important to do immunodepletion not only of plasma, but also of tumor homogenates, because there's still a lot of blood-derived proteins that has to be removed. So this study resulted basically in discovery of 8 proteins that are found in tumor, and we were able to detect them in peripheral blood. So how we decided what are those 8 proteins. So the list of identified proteins were filtered to discover in protein that were found in tumor but not in normal tissue. That they were then identified in matching plasma, and actually there's much more protein than 8, but only 8 showed spectral count higher in tumor than in plasma, so that they are bona fide proteins that are coming from tumor and not are coming by circulation from the other side. So basically this is the 8 protein that we found, that cadherin 5 and 11, deadox protein 23, kidney and brain protein, MI2, nuclear receptor coactivated pyruvate kinase, and vascular cell adhesion proteins. So those are the proteins that we believe that are tumor proteins based on our bioinformatic processing, and obviously we have to show by Western analysis, orthogonal analysis, that some of these are really in agreement with mass spec data. And we choose first to look at the cadherin 5. Why? Because the peptides which are identified from cadherin 5 are all from an extracellular domain, so that might be also a part of shedding. This is an integral membrane protein. It is involved with calcium-dependent cell adhesion and is selectively expressed in tumor endothelium in monomeric form. And we have done spectral counting on it. So even with the naked eyes, in the upper panel, you can see how many peptides we have seen in tumor, and only 2 of those we have seen in peripheral blood. The MS spectra are showing also are indicative that this is coming from tumor, because the upper panel shows the MS spectrum, which is very clear, and if you can see intensity, I'm not sure, in left corner it is E to the 4 and it is E to the 3 on the lower. So lower one is really showing an MS spectrum of a lot of noise and lower intensity. The extracted ion chromatogram also confirm upregulation or higher concentration of that protein in tumor. Then we did the Western blot analysis. We have loaded 20 micrograms of patient serum and control serum, and you can see the Western shows that it's only in patient serum. We have downloaded 30 micrograms, the signal increased. Our positive control were HUVEC cells where cadherin 5 is constitutively expressed, and then we use cell line of prostate and ovarian cancer, and that protein was not presented there. Then further, we have used serum on other 4 patients diagnosed with renal cell carcinoma, and out of those 8 proteins, we were able to show that 4 of them are present in all other serum. So that as a discovery phase, we assume that cadherin 5 is really a protein that is coming from tumor and can be served as a putative biomarker and has to be validated on bigger cohorts. Then we have done another meta-analysis, Post-hoc analysis, basically using Ariadne pathway and show what are the upstream regulators of cadherin 5, VEGF5, INT3. Some of them are known players to be involved in cancer. So, and there is also publication by Scorsia which show that selective ablation of tumor neovasculature using short-ranged alpha particles targeting the monomeric form of cadherin 5 in human colon adenocarcinoma. So basically, we have the antibody which targets cadherin 5 and deliver ionization and has shown to result in the reduction of tumor growth and increased efficiency of chemotherapy. So we also would like to say that most probably cadherin 5 is really involved in neovascular genesis, so this most probably we can say that it is a stromal marker of renal cell carcinoma rather than epithelial one. And here we also have a paradigm now that we can see in the literature that cadherin 5 really can be a biomarker and driving neoangiogenesis and can be and was used as successfully in colon cancer as a target. So next thing we wanted to see, what are the role of all these cadherins? So in our paper we have shown that cadherin 5 and 11 are found only in tumor, but when we look at the protein, that level that with overlapping protein between normal adjacent tissue and tumor, we also found that cadherin 6 and 13 were increasingly impressed based on the spectral count that you see in a tumor. So basically now we have seen all these cadherins. For cadherin 5, we have known that regulates pathologic angiogenesis. Cadherin 6 is involved in the renal cell carcinoma metastasis, and cadherin 11 is really involved in epithelial mesenchymal transition, and it's upregulated in human kidney tubular epithelial cells, and a cadherin 13 is also upregulated in growing tumor. So now we have seen that a lot of cadherins there, and luckily we found that overall upregulation of this that in literature they are known as mesenchymal cadherins in the tumor is indicative of their complex role in renal cell carcinogenesis, and the Cold Harbor Spring Review publication shown that basically cadherin switching is something which drives epithelial differentiation. So we can say that in this patient, at least as a single patient, that we can say the one part which drives tumorigenesis from stromal involvement is definitely cadherin switching that is happening there. And one also example of cadherin switching is the loss of cadherin 1, which acts as the p53, and we really didn't identify a single peptide from cadherin 1. There another molecule of interest that came up, which was not reported in this 8, which is upregulated almost 12 times in tumor, which is adipophilin, and also fatty acid binding proteins show that different kind of expression. So that shows also that fatty acid metabolism or fatty metabolism is something that may also be involved now, which is it is most probably epithelial primary cancer cells. So in 2013, Mayo Clinic researcher, they found that this adipophilin is really confirmed as a sensitive urine biomarker for early detection, and we have found it in tissue. So for us, we have then run a Western to show, and we bought just commercially available renal cell carcinoma and normal tissue homogenate, and the Western confirmed upregulation of that protein in renal cell carcinoma. The other fatty acid binding proteins that are involved in tumorigenesis, and it was found that Fab5 its clinical significance has been evaluated in ER/PR negative breast cancer. Gene expression microarray analysis revealed significant correlation between those fatty acid binding proteins in ER/PR negative tumor. So that was demonstrated by tissue array. So also Fab7 is also overexpressed, and it was also found that is found in renal cell carcinoma tissue, but not in normal tissue, using reverse transcriptase polymerase, its genomic analysis. So now we are building a story, seeing that we have cadherin switching and we have some alteration and fat adipose metabolism. And also we found recently that the adipophilin, it is the first on this table, is used as in vaccine, so it is even immunogenic for a treatment in phase 2 clinical trial of renal cell carcinoma. And publication by Straub in 2010 showed really that this adipophilin is coming from the droplets found in tumorous cells, proper epithelial cells. So we can conclude that at least for this patient, this renal carcinoma is driven by two pathways. It's cadherin switching and adipose neoangiogenesis related to the tumor growth. And now, obviously, one cannot make so many conclusions just based on the N of one patient, which is N of 1. So we really wanted to see, is there any way that we can further do a meta-analysis? And we actually found in a publication that by Toon et al., they have used 10 non-metastatic early stage renal cell carcinoma tumor and patient matched normal adjacent kidney tissue. So they have used Affymetrix platform, and they find 33,000 well-substantiated human genes, of which 13,000 were really used for final analysis, and changes twofold and more were considered significant. So what they found out, their results said that basically on the pathways that are driving these 10 tumors, it is carbohydrate metabolism dysregulation, loss of normal renal function due to EMT-driven epithelial dedifferentiation and adipogenic transdifferentiation. So this cross-validation at the messenger RNA level was performed by qPCR and also by immunohistochemistry. So this is well-substantiated study, and we wanted to see how our data coincide with this analysis. Here, this joint analysis data revealed the deregulation of carbohydrate metabolic pathways, and those which are underlined in red were also identified in our proteomic analysis. So obviously we couldn't identify every gene due by qPCR because proteomics is still under sampling, but this analysis for aquaporin and the other proteins listed in this table are really in agreement of expression shown in heat map using genomic approach. So we have very good correlation, at least for this first pathway. So the proteomic analysis also revealed the upregulation of gamma amylase and adrenomedullin in parallel with the downregulation of fructose biphosphatase and aldolase B in the tumor. So this finding is showing that our just N of 1 study is, at least in this compartment, in agreement with the genomic study. We obviously didn't discuss the protein which are differently expressed, but we wanted to see what is the congruence, is there any similarity to those two. So they have done the immunohistochemistry, and the proteins that are marked with red are the proteins that we have also identified in our proteomic study, showing the same differences in expression. So taken together, the differences in expression levels of aldolases between tumor and normal kidney observed at the gene and protein level are consistent with the substantial alteration of carbohydrate metabolism in renal cell carcinoma. This finding allows us to hypothesize that switch from isoenzyme B, prevalent in normal kidney, to isoenzyme A and C, predominant in carcinoma, is consistent with simultaneous glycolysis pathway activation and gluconeogenesis pathway deactivation. This finding we have presented in a publication. This is now in press, is accepted in Biomarker in medicine. The another aspect of that study, genomic study, is showing that EMT is a major process driving renal cell carcinoma tumorigenesis. So here is the findings by Toon et al., and also underlined in red are the proteins that we have also identified, and they are matching genomic finding. Obviously, vimentin we also found as overexpressed. The only difference is N-cadherin. We didn't find it upregulated in tumor, but other cadherins that they had mentioned before are also in line with this transition in tumorigenesis, which is driven by EMT. So the overall upregulation of these mesenchyme cadherins in the tumor are indicative of their complex role in renal cell carcinogenesis, and this is another finding of genomic study that is in sync with our findings. Then we obviously look at the genomic and proteomic data confirmed activation of lipogenic pathway, which is adipophilin is a prominent molecule. So we feel very good, encouraged and excited to see that proteomic and genomic data go along. So consistent with proteomic and genomic analysis, observed changes in expression levels of fat proteins and adipophilin may be really considered and renal cell carcinoma specific, because renal cell carcinoma has really the highest expression of adipophilin in a normal tumor. So what is important? How do we see our approach, and how one should do it? So we really do believe that if one would like to look at the tumor proteins in peripheral blood, it has to use combined tissue blood analysis in cancer biomarker discovery phase. That should be done concurrently. All samples has to be analyzed at the same time: normal tissue, tumorous tissue, preoperative plasma. It has to be done on the high accuracy and high mass measurement instrumental platform, and immunodepletion should be done on all of four. So those are the important things that we believe for successful biomarker research. And I think early studies that use very simple experimental design, that use low resolution instrument, really didn't help too much the cause of proteomics in the context of clinical research. So I would like to ponder a little bit more on immunodepletion, which we do believe that has to be done on tissues equally as in plasma, because we do know that paradigm in the blood, the 22 most abundant proteins constitute 99% of total plasma protein, and the dynamic range of protein concentration exceeds 10 orders of magnitude, and then it's really immense complexity of this. So this is why we do believe that concurrent subtractive expression profiling of tissue of blood specimen on individual basis is important, because when you do the samples from the same individual, you annulated the differences which are found in human population proper, because different people have huge differences, but biologically those differences are not relevant. This is why the samples has to be from the same sample. And then if we do tissue directed, we basically, tissue is still less complex than blood, then we move from the less complex system to the more complex system. And depletion is extremely important, as well as multi-dimensional liquid chromatography that we use, strong cation exchange, and high-resolution instruments. So obviously, when we did the first depletion of human tissue, we followed why we did it, because tissue, this slide shows how complex tissue microcirculation is. There are capillaries, there are venous blood, arterial blood, and then also interstitial fluid, and also lymph capillaries. So tissue is full of blood, and blood-derived protein definitely compromise tissue analysis. So this slide, which we show here, is really depicting that in the line 1 and 2, you can see the tissue homogenates of normal tissue and tumorous tissue, and also with the serum of the patient and standard. Then on 3 and 4, we can see that after depletion, the big thick albumin albumin band is removed, and it is almost similar. On the another panel, we can see serum which has been depleted. So basically, that tells us that, and we are really making a case that if one would like to look at the clinical tissue samples, tissue homogenate has to be depleted. So we have tested that in an adipose tissue, and what you see in this diagram, it is extremely important that we have used low resolution and high resolution instrument without fractionation. And if you look at the first 3 columns, the column D is depleted, digested, and methanol. What we have also tested, different buffers. So first 3 columns are using high resolution mass spectrometer with proteins that are depleted, that are protein sampled from tissue non-depleted, and digested in different buffers. One can see that in depleted sample, albumin was 53rd, and in non-depleted is 1st and 3rd, and that the overlap, we have run everything in duplicate, shows that is about 63% or almost 70% on average for high resolution instrument. In a low resolution instrument, overlap is only 18%. Although you have more identified proteins and peptides, that tells you that the false positive rate is huge. So that tells directly that if one is contemplating doing clinical proteomics or any kind of analysis of proteins with very complex mixture, low resolution ion traps are not way to go. And then the last 2 columns, we have run then strong cation exchange in depleted and non-depleted, and the situation become even worse. The albumin is 3rd in non-depleted, and it is 427 ranked on depleted. So basically, we have shown that even in depleted tissue samples, we have almost 30% more protein identified, which is obvious when you do the fractionation. So that tells us, and that tells us that one has really to do use mass spectrometry of high resolution, has to deplete both tissue and blood samples. So now I'm closing it. So what is the translational aspect of clinical proteomics? We have shown that we can look at the tumor proteins, in the detect proteins in blood. It is important that that can result in something called cancer proteome atlas, that the clinical sample identified, analyzed in this way, can really show what amplifies for different tumors. And then besides, which is extremely important, that can really be crucial for biomarker discovery and drug targets discovery. So now, in conclusion, I would like to say that we have developed mass-based clinical proteomics pipeline and demonstrated its capability to detect tumor proteins in peripheral blood. Second, this concurrent profiling or in parallel profiling of tissue and peripheral blood specimen on individual basis is critical for mass-based biomarker research, allowing for better characterization of tumor phenotype and pathways implicated in pathology under study. Depletion of abundant blood-derived proteins from tissue specimens should be routinely used in mass-based clinical proteomics, along with depletion of the plasma and serum. To increase dynamic range and proteome coverage, it is critical to use multiple dimensions of separation along with high-resolution accuracy mass instrumentation. So this developed technology can be used along with genomics and metabolomics for integrated analysis of clinical specimen in the context of cancer biomarker discovery. And the results from our proof of principle study, as well as the results from similar succeeding investigation from Steve Carr group and Schneider group, suggest that small number of samples may be sufficient in discovery phase, providing that initial finding can be cross-validating using orthogonal immunoassay, and then from there one can develop some multiplex ELISA assay and test it in a high throughput manner in huge patients cohorts. So I would like just to close with acknowledgments. Dwight Nisley is our Director of Cancer Research Technology Program, and to thank our collaborators from Canada, Ali Karzan and NCI, Marston Linehan, P. Wei Rong, and Bradley Starr for your summary. From Frederick National Lab, beside myself, it is King Chan, who is an exceptional separation scientist. Brian Luke is our statistician. Daru Prieto is affinity depletion specialist, and Xiao Ying is a mass spectrometrist. So I would like to thank you for your attention, and I can take some questions if you have any.